Alcohol is a drug that has been used for thousands of years by many cultures. Yet alcohol affects different people differently. Its primary target is the brain, and it has to follow a path to get there. When ingested, it moves through the GI tract into the bloodstream and gets carried to the major organs all over the body. Ultimately, a person's response to alcohol will depend on how their organs interact with it. For example, the liver is the major organ that metabolizes alcohol. But not everyone metabolizes alcohol exactly the same way. One reason is that some people metabolize alcohol more quickly than others. To learn more, let's go inside the body and see how this happens. When a person drinks alcohol, it travels from the mouth down into the gastrointestinal tract. Take a look at the organs that make up the GI tract.
Look at that.
Cool.
You can even move around the man. From the back, notice the blue vein linking the GI tract to the liver. This is called the portal vein. All the capillaries from the GI tract funnel into the portal vein to carry blood to the liver.
No, I see it right there.
Okay. Are you ready to follow some alcohol molecules into the GI tract? Let's see what happens to them.
Oh, he's drinking the beer. Whoa.
From the mouth, the alcohol enters the pharynx and then moves into a long muscular tube called the esophagus. The esophagus is lined with cells that secrete mucus, which protects the esophagus from the irritating effects of alcohol. Some people may actually feel a burning sensation here. Leaving the esophagus, we reach the stomach. A small amount of the alcohol enters capillaries that line the stomach wall to gain access into the bloodstream. But most of the alcohol is absorbed into the bloodstream through the small intestine. As we move with the alcohol molecules throughout the stomach, you'll see an opening to the entrance into the small intestine. Normally, the alcohol molecules will go right through the opening without any problems. But if you've just eaten, a sphincter closes the opening so that the food can be digested in the stomach. This means the alcohol won't get into the small intestine for some time. The slower the alcohol gets into the small intestine, the slower it gets into the bloodstream. The small intestine has a really rich blood supply. So when alcohol is in the small intestine, all of it moves into the capillaries lining the walls. Its quick absorption is also due to its structure. The kind of alcohol that we drink has only 2 carbons and is called ethanol. Because it's so small, ethanol can diffuse easily through biological membranes into the capillaries.
Wow, there it goes.
Once ethanol gets into the intestinal capillaries, it moves with the blood to the portal vein, which terminates in the liver. So the alcohol coming from the GI tract goes to the liver before going anywhere else in the body.
Is that the liver back there?
Oh, yeah.
We are now in the liver. The portal vein brought the alcohol here, where it then diffused out of the liver capillaries. The alcohol is about to enter the major liver cells called hepatic cells. In the hepatic cells, the alcohol gets metabolized or changed into another compound. The metabolism reaction is called oxidation, which requires the help of several other molecules. Let's go into the hepatic cell to see how the metabolism of alcohol proceeds. As alcohol enters the cytoplasm of the hepatic cells, it encounters enzymes that metabolize it. The enzyme responsible for metabolizing alcohol is called alcohol dehydrogenase, also known as ADH. You might wonder why ADH is even present in our bodies since alcohol is not normally present. It turns out that we do generate different types of alcohols, and the ADH can metabolize those as well. ADH is a protein. It has two identical subunits, and one is depicted here using a ribbon diagram. The curves and arrows indicate structural properties of the enzyme. However, to highlight specific features of ADH, we'll cover this ribbon diagram with a skin.
Cool.
Looks like
One important feature is the pocket in the middle of ADH. It's called an active site, and it has many hydrogen and oxygen atoms that participate in hydrogen bonding. Ethanol metabolism occurs here in a reaction called oxidation. Now, if you look closely, there's a molecule held in the active site of the ADH by hydrogen bonds. Some of these are shown in green. This molecule is called NAD, and it's shown in pink. Because NAD helps ADH metabolize alcohol, it's called a coenzyme. Let's take a closer look at the NAD.
Oh, here it is.
At one end of the NAD, there's a six-sided ring that's positively charged. This positive charge allows the NAD to accept electrons from other molecules in chemical reactions, including ethanol oxidation. Keep your eye on the big black atom on the NAD near the positive charge. It'll be important in understanding oxidation.
I think it's a carbon, that black one.
Now take a close look at the ethanol molecule. It has two carbons in black with several hydrogens attached. These hydrogens are shown in white.
Kind of looks like a
Notice
puppy
at
dog.
one end of the molecule, there's an oxygen in red. It's attached to a hydrogen atom, forming an OH bond. The oxygen is electronegative. This means it pulls nearby electrons toward it, making it more negative. The pull by the oxygen causes the attached hydrogen to be more positive and weakens the OH bond. In a few minutes, you'll see the importance of this weakened bond in the ethanol oxidation reaction. The oxidation of alcohol involves the transfer of electrons from the alcohol to the NAD. But they have to be close to each other and in the right orientation for this to happen. The enzyme ADH is crucial in providing these conditions for oxidation. Let's see how the ADH accomplishes this task.
cool.
Press the blue button.
All right, here we go.
Rather than just showing you an animation, we're handing the controls over to you. See if you can get the NAD and ethanol into the active site in preparation for the oxidation.
All right, let's give it a try.
Okay, so you're gonna grab one
This
of the
one.
molecules.
Right, and this one,
Is that the NAD?
well, let's see what it is then. Let's get it in there.
You've just placed NAD in the active
Yes.
site of enzyme
Nice.
ADH. NAD fits right into the active site and is held in place by several bonds to amino acids lining the active site. These bonds are shown in green, and they help stabilize the binding of NAD to the enzyme. Do you remember what type of bonds these are?
Oh. All right. What do you think?
If I recall correctly, it should be hydrogen bonds that hold
I
these.
think that sounds good.
There we go.
Yes, the NAD is held in place by hydrogen bonds, and it will serve as an electron acceptor in the oxidation reaction. Now, place the other molecule in the active site.
All right, so I guess
Okay.
this is the only one left.
Ethanol, right?
Should be. It's the, well, puppy-looking one.
Huh.
Let's place it in. Place it around here, right?
Yeah.
You've just placed ethanol in the active site of enzyme ADH. The positively charged zinc is attracted to ethanol's oxygen, while the hydrogen attached to ethanol's oxygen also forms a hydrogen bond with a nearby amino acid. Both bonds help stabilize the binding of ethanol to the enzyme. As you can see, the active site of an enzyme holds ethanol and NAD close to each other in a specific orientation. The molecules that bind in the enzyme's active site are called substrates, in this case ethanol and NAD. So you've just assembled an enzyme-substrate complex. Once the enzyme-substrate complex is assembled, the oxidation reaction can occur.
Cool. I guess we continue.
Yep. Press.
Okay, let's look in some detail at the oxidation reaction. Ethanol oxidation will occur in two steps. First, the ethanol transfers a hydrogen atom to ADH. And then, the ethanol transfers a pair of electrons to the NAD. Now direct your attention to that highly electronegative oxygen on ethanol. It pulls strongly on nearby electrons, making its attached hydrogen partially positive. This uneven sharing of electrons weakens the OH bond and allows the attached hydrogen to be pulled away easily. Try pulling that hydrogen off the ethanol.
All right.
No one. Yeah.
This one.
Yeah.
Watch the sparkles. They represent the electrons originally shared between the hydrogen and oxygen. As you pull the hydrogen away, the electrons move toward the oxygen. When a hydrogen loses its electrons, it's called a proton. Now that ethanol just lost a proton, the extra electrons left on the oxygen form a double bond with the nearby carbon. The extra electrons also give ethanol a negative charge. In the second step of oxidation, the extra electrons that formed that double bond violate the octet rule. Remember, atoms like to maintain the perfect 8 electrons, so the ethanol must get rid of the extra electrons. It does so by losing another hydrogen. Now, when this hydrogen leaves with a pair of electrons, we call it a hydride. Where do you think the negatively charged hydride will go?
You think something to do with the positive charge here?
Yep.
Well,
It's a good guess.
As you can see, the electrons that make up the carbon-hydrogen bond, shown in sparkles, are removed along with the hydrogen atom. The negatively charged hydride is attracted to the positively charged ring on the NAD and proceeds to bind to one of the carbons on the ring. NAD has gained a hydrogen and now becomes NADH. So, to recap, ethanol loses a pair of electrons and is oxidized, while NAD gains a pair of electrons and is reduced. Oxidation and reduction are actually two opposite sides of the same reaction, and they always occur at the same time. Before we move on, let's watch the entire oxidation reaction. The
There it
NAD
is.
enters the active site and is held there by hydrogen bonds. This happens naturally in the liver. Then, ethanol enters the active site and is held by the zinc. The ethanol now loses a proton to the ADH enzyme. Then, the hydride is transferred from ethanol to NAD, completing the oxidation reaction.
Cool.
All right.
Now you get to be the expert. See if you can drive the entire reaction yourself. First, try to assemble the enzyme substrate complex by finding the right molecules in the cytoplasm and placing them in the active site.
Okay.
Let's pick this one.
This one?
Yeah.
All right. What was this one called? Something like NAD? Something like that?
Yeah.
All right. Let's see. Goes in something like that.
Sorry, you've selected an NADH molecule. Notice the extra hydrogen highlighted here in sparkles compared to
There's two of them
NAD.
up there.
The
Right.
NADH participates in the opposite reaction called reduction. Try again.
Okay, you get to pick this time.
All right. So that was.
I don't think this one.
Yeah, that's got 2. Well, we could try this one over here.
Okay.
It's got 1. No, it's not
Might
fitting.
help if you play video games.
Yeah.
Great! You've selected an NAD molecule. The NAD fits right into the pocket and is now held in place by several hydrogen bonds to amino acids lining the active site. Now, place the other molecule in the active site.
Well, the other one
Okay.
is
The other one's the ethanol. I know that.
Right, and that's one of these small ones, right?
Yep.
Oh,
The
there's one
puppy
right-
dogs.
There's one right here. No,
Is it
I don't think
oriented
goes
right?
in maybe
You're-
the other
I
way.
I think you're close.
Yeah.
You have
there we go.
selected an ethanol molecule. It binds to the zinc and an amino acid, which orients it into position. Well done! Both NAD and ethanol are now bound to the ADH active site, forming the enzyme substrate complex. Oxidation is now ready to proceed.
Well, that was not all of it?
No, you just got the molecules in. Now you've got to get the atoms to move in the right.
Right, yeah.
Now, complete the oxidation by moving the correct atoms to their destinations.
All right.
Okay.
So let's see.
So,
Let's see what our options are here. Well, there's 3 of those. Oh, I wonder what this one does.
Good try. This hydrogen could have served as a hydride, but since it's on the far side of ethanol, it's not close enough to feel the pull from the positively charged NAD.
Oh, well.
okay, yeah, I think that proton goes first.
All right.
Correct. You've selected a proton. As you move the proton away from the ethanol, its electrons move toward the oxygen. The electronegative oxygen weakens the OH bond and allows the proton to be easily pulled off by the ADH enzyme. What happens next?
Well, then there's that.
Right, that's your only other choice.
Right.
Okay, this hydrogen has a weakened bond with the carbon. It leaves with a pair of electrons as the hydride. Correct. The negatively charged
Very
hydride is
good.
attracted
Yes.
to the positively charged NAD, forming NADH. Bravo! You've just oxidized ethanol successfully. By the way, the ethanol has been converted into a molecule called acetaldehyde, which is free to leave the active site.
There it is.
All right. the acetaldehyde now. It's not an ethanol. All right, let's go on.
But this is not the end of the story. It's actually the rate of ethanol oxidation that helps determine how people react to alcohol. Some get intoxicated, and some get sick. So let's explore how this can happen. When someone drinks alcohol, thousands of ethanol molecules reach the liver to be oxidized. But there are only a finite number of ADH enzymes present. This means that the ethanol molecules must wait their turn to get oxidized. Another oxidation reaction can't take place until the NADH comes off the enzyme. Then the process can start again. As you'll see, it's critical how fast the NADH comes off the enzyme.
So that it's empty inside now.
Yeah.
Ah, right.
It all has to do with the number of hydrogen bonds holding the NAD to the enzyme. This varies among different people based on their genetics. In some people, the active site of ADH contains an amino acid called arginine. Look at the purple-colored region of the active site. It highlights the arginine. Notice the two hydrogens on arginine that glow. These two hydrogens form hydrogen bonds with the NAD. Once the NAD is bound, these two bonds make it hard for the NADH to come off the enzyme. And the longer the NADH stays bound, the less ethanol gets oxidized over time. What do you think happens if less ethanol is oxidized?
Oh, we have to pick.
All right. So let's
Our
figure this
choices.
out.
So if less ethanol gets oxidized,
Less ethanol gets oxidized. That means more is left
in the
unoxidized and would increase the amount of alcohol in your blood, right?
right? Yeah,
Oh.
I'd pick that one.
All right.
Right, and this causes intoxication. In fact, people with the gene that codes for this form of ADH are at increased risk for alcoholism. The form of ADH in front of you is prevalent in Caucasians of Western European descent. Now, look at another form of ADH, which is prevalent in some Asian populations. In this form of ADH, the highlighted amino acid is now histidine. Look closely. It has only one hydrogen available to bond with NAD, making it relatively easy for the NADH to come off the enzyme after oxidation. Compared to the previous example, more ethanol is oxidized over time. What is the consequence of the rapid ethanol metabolism?
So this one only has one hydrogen bond, which means faster
Metabolism.
ethanol metabolism. More is broken down, so
You get more acetaldehyde. That's the
Right,
bad.
that's
That's
the
the bad
pro-
stuff.
right, that's the product.
Yes, the faster reaction rate can cause an increase in the blood levels of acetaldehyde, which is a toxic molecule. The body has the ability to get rid of it through further metabolism, but if acetaldehyde is generated too fast, it causes a flushed face, rapid heart rate, nausea, and headache. This sick feeling is enough to cause some people to avoid drinking alcohol, reducing their risk of developing alcoholism. Okay, now you can see the reaction rates catalyzed by both forms of ADH side by side. Based on what you've just learned about ethanol metabolism and how it affects a person's response to alcohol, can you identify which enzyme is associated with what type of response? The colors are gone, so watch closely.
they go in at the same time now, and alcohol is processed, but
That one comes off
comes out
faster.
first.
Right.
And this one's just
This
in
is
there
pretty
forever.
slow. That's pretty slow.
Mm-hmm.
All right.
So slower rate, more alcohol in body means intoxication.
Yes, I would pick that one.
All right, which means this one over here
Sick.
leads to sickness.
You're right. The slow rate of ethanol oxidation causes ethanol to build up in the bloodstream, increasing the level of intoxication. As you can see, simple structural differences in ADH caused by slight genetic variation can cause drastically different rates of alcohol metabolism. However, the reaction rate of ADH alone does not fully account for one's response to alcohol. A second enzyme called aldehyde dehydrogenase helps metabolize or get rid of the acetaldehyde generated by ADH reactions. And like ADH, this second enzyme has structural variations due to the small differences in the gene that codes for it. So different enzyme structures lead to quite different rates of acetaldehyde metabolism and the amount of accumulation of this toxic metabolite. Ultimately, whether or not a person gets intoxicated, gets sick, or neither after drinking alcohol has to do with one's genetics and their environment. You've just encountered one example of how a simple genetic difference controls the rate of alcohol metabolism, contributing to one's susceptibility to alcohol use disorders. Ongoing research shows that there are many other examples of genetic differences that can contribute to the likelihood of alcohol problems.
You're standing on the nucleus.
Ooh, nice.